Hello, so this is a video. Yo,
Hello,
hey man,
how you doing?
I'm good. How are you?
Alright, not bad.
Cool.
I've been looking at these parts that you have on your website there.
Oh, yeah. Which ones in particular?
Oh no, like I pretty much have added like everything that you have there that's like in stock and stuff, and
Mm-hmm.
that's like needed just for gaming that you were talking about.
Oh, what? Oh, just yeah. You don't have to have like anything um like that crazy. It's just the GPU that is the most important part. When you have the graphics card, a good graphics card, everything else. Uh, I mean, like that's the thing that will enable you to like be able to game and stuff.
Yeah.
Yeah, you don't need like a 16 core processor or anything like that, or as much
I,
RAM as I have, or any of that other stuff.
I mean, honestly, I'd still do the 16 core with the RAM.
Uh, Wait, what?
It's like, why not? I would still do the 16 core with the RAM thing.
Uh huh. Oh, cause I have like 64 gigs of RAM, which you don't need like at all.
Yeah, yeah. No, because
Uh,
I got the Mac. Like I think like 16 would be enough. Maybe 32. I think the 32 would probably be more than enough.
yeah, it depends on how much you like have like things open simultaneously. Usually the Macs really kind of are stingy with their RAM, and they don't let you add more. Like they don't let you add more yourself. That is, so usually have like a model that has like 8 gigs of RAM and then a model that has like 16, and it's like double the price.
Yeah,
which is pretty.
mine is let's see. This Mac, yeah, I have 8 gigs for RAM here.
Yeah. That's usually what I see in like a lot of Macs, and that's really like on the on the low end, because like yeah. But for instance, like on like my ThinkPad, like you can upgrade the parts yourself. So I would just get like a ThinkPad for like $200 and just put in the parts. Like, you know, on my ThinkPad I have like 20 gigs. But yeah, like 16, you know, is definitely good. In terms of like the processor, yeah, you don't need it like that many cores at all. Because even with mine, like I'm only using 8 of my cores right now. I usually like leave it at 8. So I'm not even really using it. Like the more cores are better if you're doing a lot of multitasking. So you have, you know, individual cores for individual tasks. But if since you're using it for like, you know, to game, then you want it. Yeah, it's not going to be that much. It's not going to really change things that much. It would be better to have like fewer cores but a higher clock rate, like a higher, you know, frequency. Like right now.
Yeah. I mean, at least with the Macs, I mean, I basically just only use Macs for like school and work stuff.
Yeah.
So like that's why I don't go crazy off of that. And this is a 2018, and the new ones that have come out are way better and stuff. Like they offer even i9 inside of them now. And like 16 RAM. I even think one of them has 20.
Yeah, like Macs have always been pretty late or yeah, like stingy with the RAM, because the ThinkPad that I have is from like, I think it was like, the thing with ThinkPads is that they basically last forever. Like that's how good they are. So you can get, here, one sec, T40S, release date. Yeah, from 2015. So the ThinkPad I had is made in 2015, and you can add 20 gigs of RAM to it, like yourself. Yeah. So, like, that's like usually what I use, like when I'm not on like my desktop. And I had like just a ton of ThinkPads because, yeah, because you could just get one for like $200, and they pretty much, yeah, they last forever. They're really, really well built. And I would just, you know, put in the RAM myself, put in the hard drive and stuff myself. So, yeah. So basically all you would... And then when it comes to a hard drive, like you would... I would recommend getting like an SSD for your operating system and for putting like your games on. Because whatever you're running, you want it to run on like a solid state drive, because it's going to have a much faster like read speed. And then like a regular hard drive for storage or whatever.
Yeah. A 12 terabyte hard drive is awesome.
Yeah, but like, are you gonna use like that much?
I mean, I don't know. I mean, you know, on my PS5, I run a lot of sports games because that's how it runs. But yeah, I've never really liked playing shooters on it.
Yeah. I mean, like,
so I mean, I would be running
12 terabyte kind of thing is if you're storing, like for me, like the ones that are that big, they're for surveillance, like, you know, camera footage. And, you know, I basically have, like, tons and tons of, like, movies and videos and you know, things like that I have. So that's why I have, like, you know, a bunch of, like, regular high-capacity hard drives to store things on. But for games, like, you're going to want to put them on SSDs anyway. So... just because you're running the game, like, you know, and you want it to be on a faster type of drive, like, because a regular hard drive is moving parts. It's like a spinning disk, you know.
Yeah.
And and then the SSD, it's just it's solid state, so it has no moving parts. It's much faster, basically. So like, yeah, I would say like get like you know, any type of like CPU. Look for like faster clock rates as opposed to the number of cores. And but yeah, it all really starts with the
GPU.
yeah. I see an RTX 3060 for 779. Oh wait. Okay. Yeah, I mean like, you know, it's supposed to be like 400. It's 400 at Best Buy, but it's out of stock. 400 from the manufacturer out of stock, and then 779 at Amazon in stock. So, like,
Okay.
yeah. So
which one?
I would just
Who was
keep.
it by?
What's up? Oh, GeForce.
Who was it by? G4.
Yeah, the RTX line is what you want, because I mean like it's basically like this awesome line of graphics cards that they call tensor cores, because so like when you do like deep learning, you deal with
Can you send the link to that over really quick?
yeah. Here, I'm gonna share.
Yeah, the Amazon one. That's probably when I'm gonna do it.
You know, like I would wait for like Black Friday and stuff.
Oh yeah, no, that's what I'm doing. Yeah, I'm not doing it anytime soon right now.
Yeah.
Like I'm doing it soon, but I'm not doing it like tomorrow or something.
Yeah. Yeah. So like this is what I would do. Like, you know, go to like PC Part Picker. I always look at like over here price drops.
Mm-hmm.
So you can see price drops. I would go like last 7 days, and then maybe go to video cards. And then, you know, look at the ones that are going to have a larger percentage drop. Most of these are going to be crap, but you can go to like, for instance, like this RTX 3060. You can have like set a price alert. So like you see like here on my screen where it says send an email alert if the price drops to whichever price you want it to be, right?
Yeah.
Yeah. And yeah, and then you can see like the price history over here. So, yep, that's something you could look at. Let's see if what comes up if I just type in RTX 3080. Yeah, it's still in the. It's like in the 2000s, it seems.
Yeah, 739, 779. I see a lot here. 797. Okay. Well, I mean, if that's basically the main component, that basically works anyway.
Say again?
Like, if that's going to be the main component of, like, the CPU in total, then that's worth it.
What's the main... Oh, you're talking about the GPU?
Yeah. Because, like, my budget is, like, 10... oh, not 10, Jesus.
Like 1,500?
1,500.
Yeah. So I just went to, like, browse products by video card, and I highlighted all of the RTXs, like RTX 2060 through 3090. And, let's see, I can sort it by price. So let's see, we have various 3060s here for, you know, 750 and up, essentially.
Yeah.
It's crazy how the 2060 is like more expensive, and the 2080 is like even more. It makes no sense. It's some weird supply demand thing. The 2080 is what I have. Yeah, and you know, there's also like benchmarks, GPU benchmark. Like that's what, like I kind of like look at first. Like for instance, like high end. Well, it seems like the Radeon is doing pretty well, but you see this over here, what I'm looking at?
Yeah.
So this is essentially like, you know, how I would choose like what to use. Like I would look at the benchmarks and I would go from there. So essentially, where is this? I mean these prices are crazy now. In like that first thing that I sent you, which I had like my thing, or where I talk about setting up my computer.
So what type of processor would you suggest?
Just like one that is not too expensive, I suppose. That.
And by who? Ryzen?
Oh well. I mean, it could be whichever you want. It could be Intel. It can be. I mean, I chose the Threadripper because it just was a really good like deal.
Yeah, at the time it was a good deal.
Yeah. I mean, like it's a pretty good deal in general because AMD usually is cheaper than Intel. And you know, I was like, yeah, I'll get that, but
So you see the chat. What do you think of this one?
one sec. You could also look at CPU benchmarks. Like first, basically, I would like look at the benchmarks, and then I'm trying to look for that post or whatever where. One sec. All right, so do you see this over here? So I would do this, like my method. I would like basically look at the benchmarks, right? So this was like back when I made this post, which was January 2019. So like, look at this. At the time, you see that the highest benchmark here is for something that's $6,300. And then the second highest one is $2,500. Third highest one, it's not listed. Fourth highest is $1,300. And then the fifth highest is $700. So basically, the difference between the one that's $700 and the one that's $6,300, right, is essentially like 6,000, like in terms of the benchmark points. But in terms of the price, it's like 1/10 of it. So I would look like I would basically do that, like if you want to look at what to get. So like if you want to look at the CPU, I would look at like, you know, the benchmarks, and I would look for basically whichever one has the highest benchmark but, you know, the lowest price. So over here I see right here. Do you see this over here?
Yeah.
So you see how so this is benchmarks like over here going down. So, you know, we have like 5,000 something, 8,000, 6,000, blah 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 blah. And then I see here, bam, like 749. I mean, that's still high for your purposes, but that's like the kind of like methodology I would use when choosing parts. Like I would look for the basically the most powerful part for, you know, the cheapest price, relatively speaking. Yeah. So and you see how like they tend to be Ryzen, like AMD Ryzen ones. Like this one 749, and then we have some really expensive ones, and then here's one 559, also a Ryzen, 724, also a Ryzen. The Xeon ones are for servers. You don't really need to care about that. Yeah. So in terms of CPU, like if you're using it for gaming, like, so this is the thing here. Do you see this over here?
Yeah, and look at the pre-build I just sent you. Has a lot of the same specs.
Let me see.
Like literally almost the exact, and it has a 3060 with. 3060 12 gig, that's the same thing. 16 gig RAM, a terabyte of SSD, which can always be upgraded.
I only see 2 Amazon links.
The second one.
Oh yeah, don't get anything that's already built, because it's just going to be more expensive than if you build it yourself. I can help you build it, by the way, but it's just going to be like essentially like more than if you just got those parts yourself.
Yeah.
Let's see.
Alright, yeah, I could definitely use your help building it because I don't know what I'm doing and I don't want to fuck anything up.
Yeah, it'll be a fun, like, learning experience.
Yeah.
Let me see. Brand may vary. Okay. Yeah, I can make like one for you on PC Part Picker. One of my friends like also asked me to do this, so like, and so this is like something that I was putting together for him. You could also look at like the completed builds and stuff on PC Part Picker. They're pretty good. I have like a build guide as well. But yeah, we can yeah. Essentially, once you find your GPU, you're gonna be good. The rest will be pretty simple. But it seems like the 3060 is gonna be a good one, good one to choose.
Yeah, the 36
in
is
stock.
the best way to go.
It's not too expensive, and the benchmarks for the 3060. But the thing is, the 3060 is supposed to be like $330.
Yeah, but they're in such like.
Yeah.
Well, that also happened because of COVID. They couldn't make enough.
No, it's because of crypto. Like the thing
And
with
also the crypto, exactly.
So graphics cards used to be just for gaming, but now they have two new functions. One, they're used for machine learning and deep learning research, and they're used for crypto mining. And the reason for that is because like a CPU is meant to do a lot of like peripheral things, but a GPU is meant to be like dedicated and do like a specific task. So both of those tasks, deep learning, machine learning, and like crypto mining, involve lots of like essentially like matrix multiplication and things like that. So essentially it's being used for that. Like, have you ever looked at like a crypto mining rig?
No, I have not.
So they're basically just like 100 plus GPUs just put together. So essentially it's like 100 or more. Like, here, look at this. You see this?
Yeah.
So
Oh wow.
So this is what it looks like. So essentially it's a bunch. Do you see this? Do you see it's a bunch of GeForce RTXs, right?
Yeah.
Here, these are 3060 Ti's. So it's a bunch of them that they're using to mine crypto. Like,
yeah.
So people who are mining crypto, essentially making crypto, are just buying up hundreds and hundreds of graphics cards, because that's what these crypto mining rigs are. There are tons and tons and tons of graphics cards. So that's why the price of graphics cards has just skyrocketed because of because of mining, essentially.
I see.
So essentially that's why that's why they're so hard to that's why there's not enough supply to meet with demand. The 3080, like, that was supposed to be like, so mine is like the 2080, and the 3080 is like the newest version of that. And that when it came out, or GX 20 no 3080. Yeah, when that came out, it was like $600-$700 right here. This was exactly a year ago, basically.
Oh, I see.
Right here. So that's when it came out, and then it's just gone up way, like, crossed like 1,000 and stuff like that. So I would look for I essentially, like, all of these 3060s over here, I would put like maybe price alerts, and you'll get like an email if it drops below a certain price. There's 1, 2, 3, 4, 5, 6, 7 of them, because they're going to be from different manufacturers. Like MSI makes them, Evja makes them, etc. So, and they're all slightly different, but not really that different. So you can put in different price alerts and stuff like that.
Okay. So I look at this one that I'm like trying to, I guess, build here. So I'll send you a link of what I end up taking. I'll only do this for 5 more minutes, then we'll get to work.
Yeah. Do you see this thing over here?
Yeah.
Okay, so this is the AMD Ryzen like thing that lets me. So these are all the cores, C through 16, but I only have these 8 running.
Okay, I see.
And it's running at this speed over here, 4.25 gigahertz. Now, like, essentially, like, look at it. Like, if I hit creator mode, right? That's if you're going to be doing things like lots of, like, video editing or something like that. If I hit creator mode, look what happens. Here, so this is, like, current. Creator mode, all of the clocks, all of the cores are going to be running if I do creator mode. But if I hit game mode, here, if I hit game mode, it's going to use fewer cores right here. Creator mode, game mode. Because game mode, like, you don't need that many. Oh, so you sent me. Well, I think you just sent me some, like.
I was sending you my link of what I had. Whoops. Well, like, what would you pick from, like, the things that they give there? Because I put, like, an i7 core over here, 8 core.
So the link that you sent me, it just says list.
Oh. Okay. No, because I was trying to do the, what do you call it? The system builder.
Yeah.
Yeah.
But, like, so look at my screen for a sec.
Mm-hmm.
So up here, it's not going to change, but the URL for the list you're making is going to be right here.
Okay. Oh, I see.
Mm-hmm.
I got it. Oh, all right. Give me 1 second. You would think 16 gigs of RAM would be enough, right?
Yeah, that's like, you know, I would, yeah. And you know, you can always just like get more, you know. That's like the good thing about
Yeah,
like
that's like the good
non-Mac
thing about it.
stuff, like, you know, and you know, eventually like over time it'll just get cheaper and cheaper and you can just add more. Because we're gonna
Yeah.
get you like a motherboard that's gonna be that's gonna let you put in like probably like 128 or 256 max or something.
Yeah.
Like when I first built mine, I put like 32, I think, or something, and then eventually I put it up to like 64.
Yeah. So hold on, I'm gonna copy the link. There you go. There's the one that I'm building. I've only put in 4 parts so far.
Mm-hmm.
But let's look at the total already. Holy shit.
Look at the what?
The total.
Mm-hmm. Oh yeah, it's already that.
Because of that video card.
Mm-hmm. Yeah, I mean that's going to be like hopefully you'll get lucky and maybe around Black Friday there will be enough for it to be it's the price it's supposed to be, which is like half of that. Because look, the price is supposed to be like $400, right?
Yeah.
But it's out of stock everywhere that it's that price, right? Except for Amazon, where it's like double
Being resold, yeah.
and and you know, so yeah, so hopefully you'll get lucky with that. Here,
Yeah.
this guy said, caught this during a Best Buy drop, immediately rushed to buy it. Glad I did, because the thing packs a punch for a $400 card. Great card if you can get your hands on it. So yeah, it's going like one of my roommates from like last year also wanted to build one, and I remember we were trying to find them like the card. It's like just yeah.
Yeah. So.
Yeah. So just wait until until you can you know secure the GPU.
Yeah.
But
Okay.
it's gonna be crazy. You're gonna be really amazed at what you can what you can do and what you can play. Like when I play games, like I play like them in 4K with like all the settings like up and stuff.
Yeah.
It's pretty and it runs at like a high frame rate. It's pretty cool. And you know, most people who are playing, they're playing on like 1080p and stuff. And it just looks so much better. Like it's really what like game like are you going to play anyway?
Like probably COD, League of Legends. I've been trying to get into a lot of that stuff.
Get Insurgency, man. That's
and
like,
I'm gonna try that.
Once you play that game, you're not gonna want to play COD. Like, it's just it's just like a game where like there's no HUD, there's no crosshair. It's like the guns and everything and the physics are so realistic, and there's no auto reload. Like if you run out, it goes like click click click. The reloads are in real time. It's just it's an awesome experience. People who play it are really cool. Also,
Hey, see, so I'd definitely be playing that with you, honestly.
yeah, man, let's do it. I play it like Yeah, and like I stream it on Twitch,
Yeah.
but on Twitch like they don't let you stream it at like a high quality, which kind of sucks. But
Yeah, I know, I've seen that, and then I see videos of it online sometimes, and it's
take
like
my second
way higher
Adderall
quality
today, so
than
I'm
what it
just over
shows on
here
Twitch.
yawning.
oh wait, sorry, say that again.
Like it's way higher quality than whatever Twitch is gonna show most of the time.
Yeah, so like here's
For any
like,
game too, because in PS5 it looks like shit when you watch it on Twitch.
yeah, so like here's like the game right here.
Yeah, Zoom's not having a good time.
Oh yeah, this game is an adrenaline rush.
So it's wild.
Yeah, so I would recommend that game. Yeah, this is like my Steam over here. Like I only have that and this Mass Effect like installed. That's like all I really play.
Yeah. So you see, I'd be doing stuff like that.
Mm-hmm. Yeah. Yeah, Insurgency Sandstorm. It's awesome.
Yeah.
Alright. Let's get to it.
Alright, let's do it. Holy shit, I might actually be able to sell some shoes that I'm trying to sell.
Oh yeah.
Yeah.
What, like, do you like collect Nikes and stuff like that?
No, I like when I was younger and stuff, I bought like LeBron shoes all the time and
Yeah.
stuff. But now they either don't fit or I hate how they look.
Yeah.
So, what do you call it? What do you call it? I was just thinking one day, I was like, hey, I should just start selling, them at this point, Because then
Yeah.
I was like, also, I want to get a gaming PC, right? So I was like, I should just start selling what I want at this point. Because I'm not going to use them anymore.
Mm-hmm.
And like, since they're LeBrons and stuff, and they're not in the best condition, but I'm selling them for like, I put the bid starting at like $22
Mm-hmm.
and whatever, and I already have something. It was like 4 days later, but someone finally put a bid on it over here
Yeah.
for $22. So like, I'll be able to sell it,
Mm-hmm.
and then I put like $50 buy now, and then $60 on the other one.
Yeah.
I mean, I bought them for like $200 at the time. Well, my dad did.
Yeah.
He was still around. He did at the time
Mm-hmm.
and bought them, but what do you call it? I don't use them anymore, so it pretty much comes out to my game.
Mm-hmm.
Yeah.
Yeah, and it's good. Like when you move over from console gaming to like computer gaming, you kind of you end up saving a lot of money because things are so cheap on Steam.
Yeah, that was
There's
the
so
other
many-
thing. Like, I want to play
yeah.
other games. Like, I bought Planet Coaster. I'm like into like those types of RPG games kind of thing.
What is that?
And like, huh?
What game?
Like not RPG. It's like it's called what do you call that game? Planet Coaster.
Mm-hmm.
Yeah, it's like where you build roller coasters and stuff like that. I
Oh,
bought it for the
yeah,
PS.
it's like Roller Coaster Tycoon.
Yeah,
I used to play that
but
when
it's
I
like
was a
way
kid.
way better than that and stuff.
Mm-hmm.
And I got it on PS5 because they had it, right?
Yeah.
And then I get it, and it can only like load up to like half a theme park from what I've seen online and stuff. Like guys who play it on PC
Yeah.
and stuff. They have like parks that span like literal like miles and miles, like over 50 plus miles in the game.
Yeah.
And then right when I build like 8 to 15 on the PS5 and stuff, it's telling me I've run out of storage for that file.
Mm.
The game doesn't let you go, like the game itself doesn't allow you to store more. And I had the storage on my system with no issue, but it doesn't let me go more
Yeah.
than to what I want. And I'm like, what the hell? I'm
Damn.
like, that's fucked.
Yeah.
I'm like, I literally bought it for $60, and here I am, like, ready to make miles of a theme park and everything, and then they go and tell me, oh, there's no more storage. I'm like, fuck you.
Do you have enough storage on your PS5 or whatever.
Yeah, the storage on the PS5 is 1.2 terabytes stock.
Yeah.
I have like almost 300 free. So, and that game only allows you to do like, what do you call it? I think like 2 gigabytes on the storage file until it's done.
Mm-hmm.
I'm like, what? Why? It's so stupid because there's a whole bunch of other games on PS5 that allow you to store way more than that.
Yeah.
It's ridiculous. Like the sports games store like literal 20 gigs of file on one file.
Yeah.
And it's ridiculous. So, anyways.
Mm-hmm.
I'm actually happy that shoe's getting sold.
Yeah.
And I'm charging $7.50 shipping, so I actually might still make profit off of that too.
Nice.
It's a little excessive on my part, but still.
Mm-hmm.
I'm trying to make as much profit. And these shoe people who buy and resell shoes and all that stuff, they got money, so.
Yeah.
I went to school with enough of these people to know that they'll pretty much pay anything.
The first time I was exposed to that was like when I was in college and I was walking to class in the morning and I was like, why is there a freaking line like wrapped around the block?
Yeah, they're ridiculous.
And then I met like lots of people like in the area that I live in now who like just are really into shoes, and one of them I kept telling to get into like trading or like investing and stuff, and he's like, I don't have any money, and I'm like, dude, like
bro,
you have like
spending money on shoes all the time. I don't have money.
Like his apartment, he has like stacks of boxes to the ceiling, and it's just like
bro, just resell them and put them.
Yeah.
And the crazy part is that most of the time you'll resell it for way more than you even bought it for if you have it in brand new condition.
Yeah, I thought that was the point. Like I didn't understand. I thought I thought that was the point, that people were buying it like as like an asset that appreciates in value. Like,
yeah, honestly. Have you seen the new shit that they've been doing with the Pokemon Oreos?
No.
Oh my god. I saw some in my supermarket and I was thinking about buying and they're still there. I was thinking about like buying 2 boxes or 3 boxes and then selling them on eBay for 20 each.
Mm-hmm.
Because people are going crazy over it.
Yeah.
Like they want Pokemon Oreos and stuff, but apparently they've become like really limited.
Yeah.
And stuff. Like I'll show you one listing and then we're done.
Yeah.
We're talking about that. Look at this. Hold on, Pokemon. Yeah, I'm literally gonna buy 3 boxes right after we finish our session today. People are selling so much, it's ridiculous. Some guy's already sold 389 of them at $15 a piece, and he bought them for like 5 or 6.
Oh yeah.
The thing is, it's because they're more available in like big name cities.
Yeah.
Because apparently, like throughout the U.S., like only a few places have them and stuff, and it's like L.A., New York, and then like maybe Atlanta or something.
Mm-hmm.
Have been getting the most shipments of it, in Chicago. So like everyone else in the U.S., and then I don't think they're available international at all. And literally like everyone in like Japan and everything has been trying to buy. It's ridiculous.
They're just cookies, limited edition Oreos. Okay.
Yeah, because people are trying to like, what do you call it? Are trying to find all the Pokemon.
Mm-hmm.
It's ridiculous. So, but yeah, after we're done here, I'm going to the supermarket. I'm buying like 4 boxes. I don't care.
Yeah.
Might as well make the money.
Mm-hmm.
And the thing that I find that people do wrong a lot is that they always like start the bid at a high price when you literally make more money when you start it at low.
Yeah, when I used to sell stuff on eBay, I would always ask like some of my friends to like when it would
Up the bid, right?
Yeah. Yeah.
Yeah, I was thinking of asking my friend Mugio up the bid to like $40 on one of my shoes,
Yeah,
and
yeah.
then that's then it can up the price, and then if it ends up getting sold to you, I'll just give you back the money, and then we can try it again.
Mm-hmm.
It's ridiculous. All right, what slide were we on? tRNA structure. What slide is that? What number? I got it.
Let me
61.
try to. Yeah, I think we were like around here.
Yeah, decoding the codons is where we are.
Yeah, we were like around here. Like, I just want to say, like over here, that there's 64 different codons, and
Yeah, there you go. That sounds about right. Yes.
yeah, and so this are sorry, this is like the first letter, second letter, third letter, and there's like I think it's the word is like degenerate or something. Like it means like there's multiple codons that code for the same amino acid.
Yeah.
And every single codon will code for one, but there's going to be a start codon, and then there's going to be like stop codons as well. So these are like the stop codons. And yeah, okay.
All right, so let's see. What should I write down from there?
Oh, just I guess. Well, I don't know. You don't really have to write anything. I guess just that there's multiple. I mean.
What, that there's 64 possible mRNA codons?
Yeah, there's 64 possible mRNA codons. There's 20 amino acids, so there's going to be more than one codon for the same amino acid. Yeah.
Yeah. Okay. So wait, repeat that one more time.
Oh, just 64 possible mRNA codons and 20 amino acids. So there's going to be, more than one codon for each amino acid.
More than one codon for each amino acid. All right.
Yeah. All right. So tRNA structure. So remember I told you it had, the RNA can make three-dimensional, complex three-dimensional shapes because it can form, base pairs with itself to form these double-stranded regions over here. And tRNA is a perfect example of that. So it has this type of clover shape.
Got it.
Yeah, and then so the anticodon is complementary to the codon. Like it looks like it says gay. I don't know why. Like there's a Y there. But anyway, yeah. So like the mRNA has the codon, and the tRNA has the anticodon, which is complementary to the codon. The tRNA is also the thing that has the amino acid attached to it. So, like here's like a picture of it here.
Got it. Okay.
Yeah. So that's like the tRNA. So, like, for instance, for glycine over here, the codon is GGA, and the anticodon on the tRNA is complementary to that, so it's CCU, and the glycine actual amino acid is attached to that tRNA.
Wait, say that one more time? Sorry.
So like over here, for let's say glycine, right?
Mm-hmm.
The codon for it is GGA, right? You see that?
Yeah.
And then this is the tRNA for it, and that's the anticodon, which is complementary to the codon, which is CCU. And the tRNA, like its anticodon matches up with the codon for glycine, and the tRNA is the one that has the amino acid attached to it. This will make more sense when we see like it in the ribosome. So, yeah. So,
Okay. Yeah, because that's extremely confusing right now.
Okay. So.
Oh wait, because G to C, right? A to G. Wait, no. No, hold on. Codon is GGA.
Oh yeah. And yeah, C to G, and then A to T, but since it's RNA, it's going to be A to U.
Yeah.
Mm-hmm.
Okay. Oh, okay. Got it. Oh, all right.
Mm-hmm.
So then that's how the codon translates to the RNA, the mRNA.
Well, the codon is on the mRNA, and it's going to be complementary to the anticodon that's on the tRNA, which is this thing. The tRNA is the transfer RNA, so that's the thing that actually has the amino acid.
Oh, okay.
Mm-hmm.
I see. All right.
So, yeah, genetic code is redundant, meaning, we already wrote that, meaning several codons can specify a single amino acid.
Okay. Well, so wait, what should I write about tRNA structure?
Just that it has an anticodon.
Okay.
And maybe to draw, like, the general shape.
Has an anticodon. And what does the anticodon do again?
Oh, it's just the thing that recognizes the codon. Like, the thing to know is just that the tRNA is what puts the amino acid on it. So I kind of like, this is like the notes from last time. So when we first talked about the 3D shape, I drew this for the tRNA over here.
Yeah.
So you can kind of draw that, and then like on the bottom over here, you can write like that's where the anticodon goes.
Okay. So it has an anticodon, which is just the thing that recognizes the codon.
Yeah.
And then what else?
And so the tRNA is the thing that's attached to the amino acid, and so it's going to play a role in making the polypeptide during translation.
Okay, this is what is attached to the amino acid, which will aid in the formation of a polypeptide.
During translation.
During translation, right? Okay, then that structure is just Alright, no, my drawing looks like a penis. And then the anticodon is on the bottom, right?
Yeah. It would be like right there.
And what's a D-arm and T-arm?
Oh, that's going to play a role when we go into the ribosome and see, like, there's like 3 different like slots essentially in the ribosome.
Okay.
Yeah, it's gonna be like there's gonna be like a slot where the first tRNA comes in and starts it, and it's gonna move to the next slot for the next tRNA, and it's gonna make a peptide bond between that first tRNA's amino acid and the second tRNA's amino acid, and then it's gonna move, and then that first one's gonna leave, and it's just gonna it'll have like initiation step, elongation step, termination step. So it'll make more sense once you see it in the ribosome with the slots.
Oh, okay.
So yeah, for now, just think of it as the tRNA. It has the anticodon, and it has the amino acid. For that particular anticodon.
I got it. Okay.
Yeah, so now there's something called Wobble. And what that basically means is that in a codon, the last letter isn't really that important. Like, it can be flexible. Like, but the first 2 letters are the ones that are important. So here's like an example of like for leucine, the amino acid leucine. The codon will be CUA, but it can also be CUG. And both of them will correspond to the same tRNA, which means the same anticodon. So, like, GAU, and this one is, it shows the wobble because G is not complementary to U, it's complementary to C. But since it's in that 3rd position, the wobble exists, so it can still, it doesn't really matter. So essentially for wobble, just write that 3rd base in a codon can be anything. It's the first 2 bases that matter.
Okay.
That are strict. You could say like the first 2 bases of a codon are strict and specific to the amino acid, but the 3rd one is flexible.
Okay. So. Okay. Why is this so confusing?
Oh, which part?
Like, I guess from the beginning, I'm not even sure.
Mm-hmm.
Sorry.
Yeah. Let me draw some stuff. Yeah, like, a lot of this stuff will make more sense as we go down the slides, because you'll see it, like, the full picture, in terms of, like, in the ribosome and stuff like that.
Yeah.
All right, so here I'll give you like an overview of essentially what happens. So we have our whatever like mRNA there, right?
Mm-hmm.
And you know, the ribosome comes in, so we have like the big subunit, the large subunit of the ribosome, and you know, the small subunit of the ribosome. They like, you know, clamp down on that mRNA.
Okay.
So over here, I'll kind of draw that. And the tRNA would come in on the large subunit of the ribosome, and I'll, you know, make that part, I'll magnify that part, so it's going to be like this. Codon, so let's say like, well, okay. The start codon is AUG. And the tRNA over here then would be UAC, and that's the anti-codon. And that start codon, it's for methionine.
Okay.
So essentially, so basically, like, you know, this ribosome, the ribosomal subunits clamp down on the mRNA, and it'll essentially like, and then like the transfer RNA will go into the large subunit, and then so this is like a magnified image of that, and it'll essentially it's gonna be like the mRNA is going to be going in this direction, like to the left, and it'll be like reading it, right? And the tRNA is going to be waiting. It's going to read it and wait for the start codon to happen, which is AUG. And the tRNA for it has the anticodon, which is UAC. And the tRNA is what, like, it's transfer RNA. So it's the step that takes the information from the mRNA and in translation connects that to the amino acids. So the first start codon is going to base pair with the anticodon of the transfer RNA for the start codon, which is for methionine, right? So, like, you know, that's going to be like, let's say, like, step 1, right? And then step 2, so, you know, AUG is, so AUG, you know, that's already been done. Let's say the next one is like, let's say like GAC. And so the tRNA for that methionine kind of goes to the left over here, and the new tRNA that's for GAC, which is for aspartic acid, I believe, comes in. And so this would be CUG with its anticodon, and that's for ASP. And so that's like the next step, right? And then the third step involves a connection between this amino acid and this amino acid. So I'll draw the same thing. So basically, from here to here, a peptide bond forms between methionine and aspartic acid over here. And then this tRNA leaves. So it would be like this. So the tRNA so essentially that start codon is for methionine, okay? And then that moves to the left to make room for the next codon, which is this, for, that amino acid. And so that comes in, and then a bond is made between this amino acid and this amino acid, and, it connects onto this amino acid on that tRNA. So this tRNA no longer has an amino acid, so it leaves. And, this is now in this position. And then it just goes and makes, the next step would just be, the next one. Oops. So like AUG happened, GAC happened. The next could be, how about glycine, CAA? So now this guy leaves, and the next one will be for CAA. So that's GUU, and this is for glycine. But it's going to be connected to these previous two. So ASP here, methionine here. So essentially what we're having here is the mRNA codons are being read. They're being matched up with the anticodon of the transfer RNA, which has the amino acid. So it comes in, and, as it reads the next codon, it's going to bring in the transfer RNA for that next codon, and it'll make a peptide bond between the amino acid of the previous transfer RNA to the amino acid of the next transfer RNA. So that is going to connect, and then the transfer RNA that essentially loses its amino acid leaves, and the next codon is read, and that's going to make the next transfer RNA with the next amino acid come in, and it's just making this chain. So it starts off with methionine here, and then it reads, so this is the first codon for methionine, and then that first codon is over there because it's already been read. The next codon is this, so it brings in the tRNA for that, which is for, aspartic acid over here. And then a bond is made between these two amino acids, and this tRNA part leaves, so it just leaves. And so, this is connected, and then the next one is read. So, you know, we have AUG already read, GAC already read, so the next codon is CAA, so the tRNA
And that's
for
where
that
it's
comes
creating
in.
that polypeptide.
Yeah. And then, you know,
Got it. Okay, so it's
keep
basically
doing-
just going and coding.
Yeah, so like it reads the code from the codon, and the transfer RNA is the thing that has the amino acid. So it looks like, you know, it basically is like the codon is CAA. So are there any tRNAs that are out there for the CAA? tRNA that is out there has glycine, and it's like, yeah. And then it comes in and it bonds to it, and it adds glycine to the amino acid chain, and then leaves. And yeah, that's how it makes the chain, essentially.
Got it. Okay.
Yeah. So yeah, it makes more sense when you see
Yeah, it's
it
way
with
easier
the-
like that. It's just coding
Mhm.
and making the polypeptide bond.
Mhm. Yeah.
And then one tRNA leaves once it's coded for, and then the other one starts to come in so it can be coded for.
Mhm.
Got it. Okay.
Yeah.
And that was way easier than what they were explaining.
Mhm. Yeah, yeah. Yeah. They'll have like diagrams like later on. It seems like because there are specific. There's like 3 slots. It's like A, P, and E. It's like, yeah. So let's see. Adding amino acids to the correct tRNA. That's the activation step.
So that's what a wobble codon is. The last one.
Wait, what? This over here is wobble. It just means that the third letter, or, you know, base of the codon is flexible. Like, it doesn't have to be perfect, like a perfect base pair.
Okay. Third base of the codon is flexible and doesn't have to be what?
Yeah, it doesn't have to be a. It doesn't have to be like a perfect. What's it like? You know, like Watson Crick, like base pairing, where, like, you know, A is to U and G is to C.
Doesn't have to be a perfect base pairing. Okay.
Yeah, yeah.
Okay.
Yeah, between the codon and the anticodon.
And it's between the codon and anticodon?
Yeah. Like, this is like, yeah.
Okay.
Mm-hmm. Yeah, because the idea is, wait. Alright, yeah, because the idea is that there's 61 codons that encode for amino acids, but there aren't 61 tRNAs. There's less than that. So that's why, you know, it's less specific, or yeah, that's why it's less flexible. Like, the same tRNA will code for this or this, basically. Okay. This is just this, like, I guess, activated step. We don't have to, like, this is essentially just saying how the, because I drew, like, the tRNA already attached to the amino acid. So this slide is just saying this is how that happens in the first place. But yeah, all you really need to know is that, I don't know, like, it requires energy. Like, yeah, of course. Okay. tRNA, okay. Genetic code translated by recognition events. Okay, so this is just, let's see. Alright, this is kind of like what I was kind of drawing, like, but that first step I kind of skipped, which is just, you know, essentially the amino acid attaching to the tRNA. So that's this over here. And then here's like how I drew it, like where it's already attached to the tRNA. And then you have, you know, the anti-codon there and the codon there, and yeah.
Got it.
Net result amino acid is selected by its codon in an mRNA. Okay. Translation has these three steps: initiation, elongation, termination. Start codon is AUG, so, you know, I already kind of put that into this diagram.
Okay.
I usually think of it as like August. And there's also like a machine gun called the AUG.
Oh, yeah.
Yeah, Insurgency has really cool like bullet penetration, like depending on like the material, the caliber, the velocity, the angle. So it's really cool when you can like sometimes like you can like shoot through like walls and things like that. I was using like a 50 cal yesterday and got someone by shooting through like this like thing of like the barricade of like concrete. It was pretty crazy.
Jeez.
One of the guys I play with is an ENT, like an ear, nose, and throat doctor.
Yeah,
it's really like cool. Like the people I met playing that game, most of them are like, you know, in like lots of like flyover states and stuff. So, and makes sense because the game, that type of game, attracts people who like know a lot about guns and have a lot of guns. And lots of like vets play that, like veterans play that game too. One guy I play with like has like one arm that like he was like blown up from like an IED. It's pretty crazy.
oh wow.
Yeah. Like yeah. One of my closest friends on there is like 64 or something, and we play with his father-in-law who's like 76.
My goodness.
Yeah. All right. Yeah. AUG. That's the start codon. All right. So this is, I guess, just
Okay. Initiation, elongation. Okay.
yeah. We can forget about this one right here. This is just kind of going a little too in depth.
Okay.
Okay, so yeah, so here's like a good picture of what I was saying about like the 3 slots that are there on the ribosome.
Mm-hmm.
So that would be like a good thing to draw. Like there's an E site, P site, A site.
Yeah. So wait, with initiation, AUG is always going to be the start codon, right?
Yeah.
Okay.
Both in eukaryotes and prokaryotes. Eukaryotes, it codes for methionine. Prokaryotes, it's like formic methionine. It's like a version of methionine that has like post-translational modification on it. But yeah, it'll always be AUG.
AUG is always the start codon for prokaryotes and eukaryotes.
Yeah.
Okay, nice. Alright, we're skipping that.
Yeah. And so this is like what I was saying about the slots. There's an A site, a P site, and an E site. So,
Yes.
yeah.
Elongation. And then draw the little thing, right?
Mm-hmm.
This is the small ribosome. Oh, okay. Got it. And then the top one is the large ribosomal subunit.
Yeah.
And then the EP and A are all different tRNAs?
Yeah, so I wrote some stuff here. So yeah, A site is where the tRNA enters. The P site is where the new tRNA and the previous tRNA meet, and their amino acids form a peptide bond.
Wait, say that one more time?
So the A site is where the tRNA enters, like it comes in.
Mm-hmm.
And then it moves to the P site to allow the next tRNA to enter. And when they reach the P site, the previous one and the new tRNA, they meet, and their amino acids form a peptide bond.
New and previous tRNA. Okay, and then E is previous tRNA exits. Okay. Got it.
Yeah, so like where I drew this, so this like, let's say this is like at the whatever A site, and then here this one is at the P site and this one is at the A site. So, this moves over. And then this is like where they're at the P, now
P site.
this one goes to the
P
P
moves
site,
to
makes
P,
that
and then P is
connection.
moved to
Yeah,
E
this
'cause
one
it's leaving.
leaves. Yeah,
Got it. Okay.
Okay.
That's pretty.
Mm-hmm.
Okay.
So, I guess. Yeah. All right. I'll just go in. I mean, this is just kind of like just saying that GTP, like essentially energy is needed for the tRNA to essentially bind into the ribosome. It's not really like, all right, yeah, whatever. Forget that for now. Let's see.
Well, this is the enzymes, I think. I mean, the different.
Wait, what's up?
P and E.
Yeah. This is just saying that, like, it requires GTP for the tRNA to bind, to the A site.
Mm-hmm.
So, but all we need to like really care about is just that's the entry of that first tRNA. Here's where the peptide bond is formed. So okay, like this is like the start codon, the first one, right?
Mm-hmm.
And then the next one gets added to the A site, and then after that, there's a peptide bond that's made between the amino acids for both of these. And then, this one goes into the P site. The one that loses its amino acid goes to the E site where it's ready to leave. And then it leaves, basically.
Okay.
Yeah. And here's, like, peptide bond formation, like what it essentially looks like.
Yeah. Basically what we were talking about already.
Yeah. This is just like going into like the actual like what it
the biostructure
of it. Mm-hmm. Okay. Yeah, they're really specific here, and I don't from like the test that you sent me, it doesn't seem like they're going to ask anything that like really that specific, but yeah, so we can go through that pretty quickly for now. Yeah, termination is so these are the stop codons. This is you can kind of see an example of Wobble over here, where these two both have UA at the beginning, but the last one is different, but they're still gonna have the same tRNA, right? So that's kind of like an example of Wobble right there. But yeah, they're stop codons, so they basically just mean that these release factors bind to the A site, and then it causes the ribosome to disassemble, like the large subunit and the small subunit to disassemble.
Okay.
And yeah, the mRNA is released. So that's when, you know, the peptide is done being made. Alright, and then mutations can cause early termination, so that's called a nonsense mutation. Oh yeah, they have it here, nonsense. Yeah. So that's just when some type of mutation happens that makes a premature stop codon, and it just causes the, like, the whatever peptide that's supposed to be, like, made to just not. Like, it just stops it prematurely.
Is that each cycle lasts 1/20th of a second? On the last slide?
Each cycle lasts 1/20th of a second, yeah.
Okay. Fast.
In other words, 0.05 seconds.
All right, ready for termination. Can you go back? Yeah. Where's that? Go back like 2 more. The release factors. What do I get? What do I copy from there?
Oh, I mean, like, yeah, I don't know, like, how in depth we have to go about it. I mean, you could just say, like, for termination, release factors. Okay, okay, all right, so for stop codons, there's no tRNA for them. Instead, there's going to be release factors that mimic tRNA, and the release factors are the things that bind to the codons and basically causes the ribosome to fall, you know, to disassemble. So, like, you know, that's why they said, like, 61 of the 64 codons code for the amino acids, because the other 3 are the stop codons.
All right. You're gonna have to say that one more time.
Here, I can draw it. 1 sec. All right, so here it is over here. So basically, like, once, like, UAG, that's an example of one of the stop codons. So once that's reached, instead of like every other codon where a tRNA comes in, a release factor comes in. And that recognizes the stop codon. It mimics the tRNA, which means that, it goes into the A site like a tRNA would, and it causes the ribosome to disassemble.
Alright, so basically once one of these codons happens, it activates a release factor. Which mimics the tRNA.
Yeah, and recognizes the stop codon. I can probably scan this for you. It's on a page from my notes where I go over essentially everything for this.
Okay.
Do you see that?
Yeah.
Yeah. In this notebook, I have everything about everything.
Got it.
Even what the D loop and T loop do and stuff like that.
Got it. Okay.
Yeah, and then these are just different types of mutations where, and
yeah, nonsense mutation, missense, right?
Yeah, I think this one is the missense one, because that's when it's something is replaced, but since there's Wobble, it's not going to really change anything. It's still going to code for the same amino acid. So
yeah,
that would be a missense. Yeah, nonsense is when it changes it to a premature stop codon. And then if there's an insertion or deletion, it's going to alter the reading frame. So that's where it messes everything up. It messes the frame, the reading frame. Then there's something here called nonsense mediated decay. So apparently, I guess it's just something that can kind of recognize mRNA that has premature stop codons, and it just gets rid of them. Could be some type of proofreading thing. Don't really have to write much on that though. What is this? Polyribosomes. Oh yeah, there could be multiple ribosomes working on a single mRNA. Yeah, I kind of drew that up here.
Working on a single mRNA.
Mm-hmm.
Okay.
And yeah, this is the last one for this PowerPoint. How some antibiotics work. So, a lot of them are going to be working by messing up the translation of certain proteins. So, tetracycline blocks the binding of the tRNA to the A site, which is step 1 basically. Streptomycin prevents the transition from initiation complex to elongation, so can't do like the peptide bond thing. Same thing with this, and this is the translocation step where it moves from the A site to the P site, I suppose. Yeah. And then this blocks initiation of transcription by binding to. So yeah. Okay, and then these are specific for prokaryotes.
Oh, okay.
Yep. So let's see. Right. Let me go to the next one.
Next one is on gene expression. Why? At least it's only 36 slides.
Mm-hmm. Gene expression, okay. Human neuron and liver cell have the same genome. Yep. But express different RNAs and proteins. A typical cell expresses about 20% of its 25,000 genes. A cell can change the expression of its genes in response to external signals. That last part is epigenetics.
Okay.
So, yeah, gene expression can be controlled at many steps. So, it can be controlled at transcription. It can be controlled at processing the RNA. So, let me ask you, what is the processing that happens between this and that?
In the RNA and mRNA?
So first, the DNA is transcribed into RNA, and then for it to turn into, the...
Well, it needs to be translated, no? Transcribed?
Yeah, so it's transcribed first to make this. And then, but certain things have to happen. Remember those 3 things? For it to turn into the mRNA? Because you remember how, only 2% of the genome actually is, genes are actually genes?
Yeah, codes for actual genes, yeah.
So in the transcription step, they transcribe everything, from the DNA, right? Including all that stuff that we don't need. So... But to make the mRNA, where it's just the stuff that we need, certain things happen.
I'm gonna feel dumb when I don't see it. Oh, what is it?
Here, maybe this can help right here.
Oh, it left. Oh, that? Okay. DNA transcription. Oh, alternative splicing, the 5' and the poly A tail.
Yeah. Because the DNA is going to have all that crap that we don't need. So, the first transcript, the RNA transcript, is going to be exactly that of the DNA. But then we're going to so what are the regions that we don't care about called? Starts with an I.
Say that again?
What are you like doing?
Just answering updating really quick.
Mm-hmm.
All right. Say one more time.
Oh yeah. So the parts of the transcript that we don't need, what are they called?
Parts of transcript that we don't need.
Yeah.
Pre-RNA, pre-mRNA.
Oh well, that's the entire thing that has the parts we need and the parts that we don't need.
Oh, okay.
So the parts that we need. All right. The parts that we need. What are those called? It's like a oil company.
Oil company. Not BP.
No. Do you remember the something Valdez oil spill?
No.
Oh, okay. What about something mobile?
Yeah.
Oil company that's blank mobile?
Oh. Exxon.
Yeah.
Yeah.
So that's the part that we need, right?
Yeah.
What's the part that we don't need? What is that called?
The intron.
Yeah, good. So basically the introns are the parts that we don't need. Exons are the parts that we do need. So when it gets spliced, it splices the exons. It splices the introns out, and so the mRNA is just going to be the exons. So it's going to be much shorter. It's not going to have any of the, junk kind of DNA. And what are the 5-cap and poly A tail for?
The 5-cap. Well, isn't that where it starts the reading?
Like the 5 end, yeah. And it's going to be like, a 5 end and a 3 end. But what's the cap and the poly A tail for? Like, what are they for? Like, the purpose?
Well, poly A tail has to do with amino acids, huh?
So the poly A tail is just... So, okay, if there's a 5 cap, where do you think the poly A tail will be?
That means it'll be on the 3 end, no?
Mm-hmm. And since it's just a part of the mRNA, and amino acids don't come into play until the tRNA comes in, right? With the translation step. Just think about, what's the purpose of having those things on both ends?
Well, wouldn't the poly A tail keep coding for more mRNA?
Well, okay. The poly A means that it's just a bunch of A, like adenosines.
Mm-hmm.
So it's not going to be coding for anything because it's just A, like constantly. But the reason for these things, though, because they essentially like add on to both ends, is to protect the mRNA from being degraded. Because the RNA itself is very, very unstable. It's inherently unstable because of its OH group that it has. But it's also unstable because the cytoplasm has lots of exonucleases that break it down. And exonucleases break it down at the ends. An endonuclease breaks it like somewhere in the middle. So like, when you make restriction enzymes to make those like DNA fragments and stuff, they use restriction endonucleases, but exonucleases are going to be at the end. So you place the 5 cap to protect that 5 end and the poly A tail to protect the 3 end from being degraded by the enzymes outside of the nucleus.
Oh, okay.
All right. So the splicing part is to get rid of like the junk, like parts of the transcript that don't code for anything. So that's what we do first. So we make it a lot shorter, and the 5 cap and the poly A tail are to protect it.
Okay.
All right. So that's what's happening here in RNA processing.
Mm-hmm.
All right. And so when it's processed, it's officially mRNA, and mRNA could then leave the nucleus through a nuclear pore and enter the cytosol, right?
Mm-hmm.
So that's the third step here. mRNA transport and localization control. So these are all just, steps that you can control gene expression. All right, and this is a pretty good schematic for like how everything kind of like works. So in terms of like really simplifying it. So see how over here at step 4, mRNA degradation control.
Okay.
Over here, do you see that?
Mm-hmm.
So this is what I was saying about like how mRNA, just like RNA itself, just like is going to be degraded by nucleases and things like that in the cytosol. Like even mRNA, even with the 5 cap and the poly A tail, because like mRNA, is a short-lived type of molecule that's only there for to be immediately translated into the protein that it's supposed to make, but it's not there for any other purpose. Whereas DNA is there to be held as information storage, mRNA is just that in between. So it's not meant to live or last. It's just meant to be made when you need to make something.
Okay.
All right. So this is a control point where it gets degraded. And then here's the translation control where it turns into a protein. And then there's a protein degradation control because the protein itself can be degraded by proteases. And then here's the protein activity control where you can activate the protein or, deactivate it.
Mm-hmm.
So, you don't have to do this now, but this would be a good thing to, draw out. And kind of test yourself being able to draw it out from memory, because then you're going to know pretty much, you're going to have a full kind of understanding of the entire kind of process and how everything works.
Got it. Okay. I'm taking a screenshot of it so I can attach it over here to my notes.
Mm-hmm. Yeah, and try writing it out also, just so you can test your memory.
Oh, no, of course, yes.
Yeah.
Well, I'm just putting it there for time purposes right now.
Yeah, of course.
And plus, it's more detailed than what I can probably write it in so far.
Mm-hmm.
Okay.
All right. So this is just how much. So each of these points can be where you control gene expression, and this just shows you how much it's controlled at each of these steps. So like it seems like the vast majority, like almost 3/4, 73%, are at the transcriptional level.
Mm-hmm.
And yeah, 8% the translation, 8% the protein degradation, 11% the mRNA degradation, etc.
Okay.
All right. Let's see. Differentiated cells contain all of the genetic information to form a complete organism. Cells can change the genes they express without altering the nucleus. Yeah. So that, I mean, that's kind of goes without saying.
Okay.
Like, yeah, you don't have to alter the DNA at all to alter, like, you know, what gets expressed.
Okay.
Okay, so okay, so this is just saying how do we determine which genes are expressed. You can identify all of the mRNAs that are found in certain conditions. So this is basically like, you know, talking about like the Lac Operon without talking about the Lac Operon, but you know, this is just it's like yeast. So like change in conditions during experiment, right? So like yeast grown in glucose deplete the glucose that it makes as ethanol is made, and then they use the ethanol as a carbon source. So that's why you kind of see the glucose here in blue just go straight down, and the ethanol go up.
Okay.
And then as the ethanol go down, the cell density goes up.
Okay.
So that's just like, since the glucose is being used to make the ethanol, you'll see as glucose goes down, ethanol goes up. And then ethanol is used as a carbon source to make, you know, things that need carbon, aka parts of the cell. So as ethanol goes down, the cell density will go up. So like first, as glucose goes down, ethanol goes up. And then as ethanol goes down, cell density goes up.
Okay.
Yeah, this PowerPoint seems to be more like about like things that can be done in terms of like research science. I don't know if it's going to be like things that you have to like write much about. Because I kind of looked through it and they're talking about like deep like microarrays and things like that.
I don't think we're even like really going into it like that. Let me check the syllabus actually too, what's in the test, so we can prioritize.
Oh yeah, and I looked through the test also. Yeah, there's
no, I mean like what it's going to be on the actual test. I mean, like the chapter.
Wait. Oh, okay.
Yeah, chapter 8 is going to be on there. All right, so this one's the last chapter on that.
Uh-huh. Wait, the test that you sent me, is that the material that's going to be on your test?
Yeah.
Okay. All right. So, okay. So this is just okay. So this is just okay. So this is an example of them identifying mRNAs. So they basically. Oh yeah. So cDNA is complementary DNA, and it's basically made from RNA. And the importance of that is that it's much shorter because it's made from mRNA, so it's much shorter. So it just has the genes that get translated.
Okay.
Yeah. So they basically make these cDNAs, which are essentially just the same as the mRNA, and they put a green dye for the ones that are in from the cells that are in glucose, and they make the ones that are the cells that are in ethanol without glucose, meaning that glucose is depleted. They make that red, and then they mix the two populations together and they incubate it in the cells, and then they make this like microarray thing, and they can look at the intensity of the red light or the green light that it gives off to indicate how much of the of each mRNA is expressed.
Got it. Okay.
And then, so like if you take a look at this over here, I'll zoom in on this.
Okay.
Okay, so like for instance here, right?
Looks
So
like Connect 4.
what's up?
Looks like Connect 4.
Yeah. So so this is just an array. Array is just a word for something that is kind of like in a grid, because it's arranged array, right? So if it's like very green, it's going to indicate a lot of mRNAs in the cells that have a lot of glucose. If it's red, it's going to indicate that there's a lot of mRNAs that are in the cells that have depleted glucose, aka ethanol, right? Yellow is a mixture of the two colors, so it's going to be like roughly equal amounts of both. And then gray means that there aren't any mRNAs there, which means that it's not expressed. So essentially that's what the array is kind of showing us. Like that's how we can figure out like if something's expressed or not. Because if it's expressed, it's going to have the mRNA, and if it has an mRNA for each, specific thing, we'll know exactly like what color it would be.
Oh, okay, I see.
Yeah, they do like these types of testing like a lot, like for the rest of like your science career and on the MCAT and stuff. So I just want you to be like kind of familiar with the idea of putting like a fluorescent tag on something to figure out how much of like that molecule there is. Because otherwise, like if I said like how many mRNAs, like tiny molecules of mRNA are in a cell, you'd be like, how you'd be able to figure that out. But that's how you can figure that out. You can add fluorescent tags, and you can evaluate the intensity of that light. Intensity means like brightness, aka number of photons. So
okay.
And so, yeah, here's like the results of a DNA microarray. So, yeah, like I said, green, glucose utilized, red, ethanol utilized, yellow, mRNAs equal for both, black, basically no mRNAs, which means that nothing's expressed. And then, by intensity, like, the less green it is, fewer mRNA for that. The more green it is, more RNA for that. So that explains like the different shades.
Okay.
Yeah.
Are we really supposed to like know this, or like what is this?
Oh, this is just like, I mean, like you don't have to like write any like much down for this. I just want you to like kind of understand the like principle of it, because you're gonna kind of like see this stuff like on a lot of things in your career, and maybe later parts of the test or something, of your curriculum or whatever.
Okay.
But yeah, it's just good to be able to like understand like like the experimental procedure kind of thing. Like if you saw like this graph, like to be able to interpret it. Like for instance, like if you see one thing going down as another thing goes up, like you do with glucose going down and ethanol going up, you know that glucose is turning into ethanol or is being used to make ethanol. Yeah. And so, okay, we can kind of move on, I guess, then. This is, all right. mRNA levels of 1,800 human genes and 142 different human tumors. Higher than average expression are red and lower than average are green. Alright, so this is kind of interesting. So, different types of cancers and different genes, right? So, red means it's higher than average expression. So, those are the areas that are going to be, like, highly cancerous. So, this is a way for us to locate the genes that are in these different types of cancer. Like for liver cancer, it'll be like this red region. So like these genes, right?
Yeah.
And then, yeah. So let's see. Yeah, each class of tumors has its characteristic spectrum of expressed genes. In this case, the UNC type yellow tumor has been judged to be a unknown. Yeah. So like this unknown whatever tumor was judged to be okay, so like for instance, let's say you have you do like a microarray of your whatever cells, right? For like let's say you have a tumor and they do a microarray on it, and then we're gonna see like what type of cancer is this. They can compare it with the types of the microarray stuff for all the other types of cancer, and it'll match. Apparently matches up to be lung cancer because its pattern of gene expression is similar to those of a series of already identified lung cancers. So.
Okay.
Mm-hmm. Transcription is controlled by proteins binding to regulatory DNA sequences. Gene expression begins at the promoter. So, remember that from last time?
Mm-hmm.
Yeah, promoter over here. Whoops. Yeah. Promoter over here. Binding site, okay, for RNA polymerase, aka where transcription starts. Alright, in selective gene transcription, a decision is made about which genes to activate. DNA regulatory sequences are used to switch genes on and off. Transcriptional regulators bind to those sequences. In the most general case, two types of regulatory proteins may bind DNA: repressor proteins and activator proteins. All right. So there's positive regulation, which are activators, and that means that if for a gene that is not normally transcribed, an activator, once that binds to it, it turns it on. So it makes it transcribed. And the opposite of that is negative regulation or a repressor. So if a gene that's normally transcribed, a repressor protein prevents transcription. And I am going to predict that they might talk about this, but for cancer in particular, there's something called like a 2-hit something, which means that 2 things must happen. A proto-oncogene, onco is, for cancer.
Yeah.
Proto-oncogene that's not normally expressed must be expressed, so an activator can bind to that. And then there's a tumor suppressor gene, which suppresses the formation of tumors. That's normally expressed, but that can be turned off by a repressor. So cancer will form from those 2 things happening, which is a proto-oncogene being turned on and a tumor repressor gene being turned off. So that's just a real-life example of what they're showing here with the activators and repressors.
Okay.
Okay, and then I guess this is just saying binding of the regulator, whether it's an activator or a repressor, binds to the major groove of the DNA helix, which, makes sense because that's a bigger kind of area. Alright, now here we go with the operon. So cluster of bacterial genes can be activated by a single promoter. And, this is what I was talking about before with, over here, prokaryotes and stuff. They can have multiple genes, polycistronic, right? So this is an example of that. Each of these are multiple genes on a single, mRNA. And it can make several different things.
Okay.
Oh, and then this is okay. This is saying if tryptophan concentration's low, then it's going to express the enzymes that you need to make tryptophan. If there's too much tryptophan, then it'll repress the genes that'll make the tryptophan.
Okay.
Yeah, here's just another picture of it. So if tryptophan is low, the the repressor will be inactivated, so it'll turn the operon on and make more tryptophan. If there's too much tryptophan, then it'll activate the repressor, which will turn the operon off.
Okay.
Alright, and this is what I kind of drew here when I compared the prokaryote to the eukaryote. What I drew here, that's kind of what it's saying here. I'm going to be right back. I'm just using that.
Okay.
Okay. Yeah. Now, just for eukaryotes, there's going to be. It just says it occurs at a distance, so it's just much, much further away.
Got it.
And then remember this entire thing, the pre-initiation complex.
Okay.
Okay. Oh, and then this is all that stuff with histones and chromatin and stuff like that.
Okay.
So that's another way that genes can be controlled, like their expression can be controlled.
Mm-hmm.
So, yeah. Yeah, essentially, like, you know, if it's too close together, it's not going to be expressed. When it's spread apart, it's going to have room for transcription factor to come in and have it expressed.
Okay.
Yeah.
So
So
how can I write that down?
Oh yeah. So yeah, generally speaking, like of course, the more condensed the chromatin is, you know, the less expressed it'll be. And you can say that if the histone is acetylated, it's going to spread it apart, which means that it's going to be expressed because it's going to spread it apart.
Repeat that again.
Yeah. So you can. All right. So you can say that. You know. Okay. So the chromatin.
Yeah. The more condensed chromatin is, the less expressed it will be,
Mm-hmm.
and then.
Oh, and then if the histones are acetylated, it's going to spread them apart, making them expressed.
Okay.
All right, and then this is just you know, condensed chromatin. You know, it's off, and when it's decondensed, it's on. Because essentially, it's so condensed that there's no room for a transcription factor to bind to it for that pre-initiation complex to form. So when it gets activated, you see this gap over here when it's decondensed, which makes room for this entire, you know, initiation complex to form, which will make, you know, the RNA, so it'll get expressed.
Okay.
A single transcription regulator can coordinate the expression of many different genes. So, okay. In eukaryotes, many genes involved in the same pathway are on different chromosomes. Let's see. Series of genes with activator proteins bound, however not efficient activation, additional transcription. Okay. Alright, I don't think we need to write anything for that. This looks like a mess. Okay, this is just whatever for a particular gene. Okay, now they're saying how is it regulated after transcription? So all the stuff that we went over just now was the transcriptional control. And now they're saying now we're gonna start after that. So now we're gonna be talking about the RNA processing control. So exactly what I said before, like, alternative splicing of pre-RNA, microRNAs, translation repressors, regulation of protein breakdown. That's all the stuff, like, all the way over here. So mRNA translation can be regulated, so that's over here. So protein and mRNA concentrations are not consistently related. It's governed by factors acting after the mRNA is made. Cells can block mRNA translation or alter how long the new proteins persist in the cell. Multiple ways to regulate mRNA translation. MicroRNAs can inhibit the translation. GTP cap, aka the 5 cap, right? On the 5 end of the mRNA can be modified. If the cap is modified, the mRNA may not be translated. Repressor proteins can block translation directly. So, okay.
This is basically reviewing what we've done already.
Yeah, it's going into like how things can be controlled at these later stages. All right, let's see. MicroRNAs. So I guess it would be good to like write what that is. It's just a very, very tiny type of RNA, around 22 nucleotides. Of non-coding RNA. Non-coding means that it's not going to code for an amino acid.
NT is nucleotides?
Yeah.
Of non-coding RNA. Alright, keep continuing. Sorry, I didn't mean to interrupt.
No. Yeah. So, those microRNAs, that can inhibit and change things. It can silence RNA.
Wait, say that one right now?
Oh yeah, so it can work to silence the RNA.
What, the RASC?
Yeah, it can make that complex. Like anytime you see complex, like the pre-initiation complex or this complex, it's just like a thing made of multiple proteins and stuff, like a machine essentially. The assembly of like a machine. But that's just like the method, whereas the microRNA is like the actual thing to like focus on, because that's the thing that causes it rather than the execution of it.
Okay.
Yeah, so you can say like microRNAs function by silencing the RNA, aka inhibiting its translation, and also causing the mRNA to be degraded. And yeah, over here you see how it says degraded by nucleases. That's what I was saying before. Nucleases are the things that would degrade the RNA.
Okay. So basically, microRNAs can create an RNA-induced silencing complex where proteins will guide the mRNA translation, and inhibit and degrade it.
That's proteins guide the microRNA to the target mRNA.
Oh, okay.
Yeah, so like essentially like you can kind of go through it like the diagram here. So like, precursor microRNA, whatever, it leaves the nucleus, right? It forms this RISC complex, which is this thing over here. And then that just searches for the target mRNA. So
yeah, okay. So basically, a microRNA will create an RSC, and then the RSC is basically when a protein will guide the the microRNA to the target mRNA, where then it can degrade and inhibit mRNA.
Inhibit mRNA translation.
Yeah, mRNA translation.
Mm-hmm. Yeah. So like, if it's not that extensive of a match, then it's just going to reduce, or inhibit translation, turning it into, the process of turning the mRNA into the protein, and then it lets it eventually get degraded by the nucleases in the cytosol. But if it's an extensive match, it has the mRNA get degraded like instantly by the nucleases.
Okay.
Sequestered means that it's like trapped or no longer. So it's almost like in war, like if your enemy surrenders or something and you like let the enemy go, that's like this over here, less extensive match. Like translation's reduced, you don't let the enemy combatants continue to like shoot you, but you don't, and you just take them as captives, like so they're taking the mRNA as captive. But if it's an extensive match, they just kill the mRNA. So that's like, if you don't want to take the enemy captive because you don't want them to like get released or whatever and then keep doing it again, you just want to kill them. So that's what happens like if it's an extensive match.
Oh man. Got epithelial cells. Oh wait, no, never mind.
Alright, so I kind of like drew it like this. So like microRNA, if it's a low match, it'll stop translation, but it lets the mRNA degrade naturally. If it's a high match, it's just going to destroy the mRNA straight up.
What does UTR stand for again?
Oh, untranslated region.
Okay, and then what does that thing say?
This right here?
Uh-huh.
Yeah, so this is just me kind of like simplifying it. MicroRNA, if it's a low match, it'll just stop translation, but it lets the mRNA be. Like it's like, it's gonna die anyway from like nucleases that exist naturally in the cell. But if it's a high match, like, high ranking, enemy combatant, it straight up destroys it.
Got it. Okay. Makes sense.
Yeah. A better analogy would be like if you're like in air combat, like, and you shoot down a plane, enemy plane, and the enemy pilot, ejects out and parachutes down. You stopped that translation process. You stopped, the creation of proteins, but you let that pilot survive. But if it's like really, really bad, you want to kill the pilot, because if you don't, the pilot can just get into another plane and then cause, wreak more havoc.
Yeah.
So that's just the difference between like this thing where you stop the translation but you let the mRNA survive because it's going to die naturally. Whereas if it's an extensive match, you just want to kill the mRNA right away. Alright, gut epithelial cells can control which bacteria live in the intestine.
Okay. That's basically like an example, I guess.
Oh, yeah. Because, yeah, pretty much, I guess. And then this is translational control, which is probably going to be the last one that they talk about. This is an example with ferritin. There's lots of free iron. A lot of free iron in the cytoplasm is harmful for the to the cells. Ferritin protects the cell by binding to free iron. So if you have low iron, this iron regulatory protein binds to iron response element in the untranslated region and blocks the ribosome from binding. If you have a lot of iron, the IRP changes shape, and remember, shape dictates function, and it's released from the IRE, which lets translation begin, and it can bind to ferritin can bind to the free iron. Oh, and then I guess they're just gonna do like one slide for each of these last two. Protein degradation control. So protein longevity is regulated. Protein content is a function of synthesis and degradation. Ubiquitin attaches to a protein to mark it for destruction. So, that's an example of a post-translational modification. Maybe write that down just because I've seen it come up in like other kind of stuff.
Alright, so tell me what I should simplify it as.
So I would say like proteins get so I wrote proteins get ubiquitinated, which means that a ubiquitin attaches to it and it marks them for destruction. So it's like, when I always use like war analogies, when like medic or something uses like chalk or something to like triage like the wounded casualties, and like, this one's definitely gonna die, so he puts a mark on it, so when other doctors and stuff come in, they're like, all right, let's not waste time on this person because he's definitely going to die. And then the ones that can be helped, you know, they don't mark like that. So it's something, it's like a, you know, something that gets, it's a mark for destruction, basically.
Okay.
Yeah, so whatever. It basically, this is like the method for it. Like, binds to a proteasome where the...
So I can write after, and it binds to a proteasome, and...
this is just...
or are they separate
the
examples?
execution. Like, you know, like, there's, like, the thing that actually causes it and the execution. So, like, here, like, the microRNA is the actual thing that causes this stuff. And the RISC is, like, just the process of it. So, you know, the thing to, like, if it's ubiquitinated, if it has a ubiquitin attached to it, it's going to be destroyed, and this is just saying how it's destroyed. So, you know, you don't really have to write it, to be honest. Like, yeah, like, it's, like, kind of saying, like, the mRNA is what gets translated into the protein, but the ribosome is how it happens. Like, one is the thing, like, the mRNA part is the important part. The ribosome thing is just, you know, the process of it happening. Alright, and this is the last one for this. Cytoplasmic localization of mRNAs. Cells need actin near the leading edge of cell for locomotion. mRNA for beta-actin is inhibited until localized to leading edge. Okay. So, like, mRNA basically doesn't do anything until it hits that leading edge, and then it gets translated into the actin, which needs to be at the edge for the cell to move.
Alright. Alright, that sounds good. So, well, alright. So I guess we'll meet next Wednesday.
Oh wait, you don't want to go over this test? Because wait, when? Tell me when your test was again.
Oh, October 4th.
Okay. Oh, that's further than I thought.
Yeah.
Okay.
Yeah, so we could just start fresh and start making a review sheet next time or something.
Alright, we can do that next time.
Yeah. Alright.
Alright.
So I'll see you, man.
Alright.
Thanks.
Take care, dude.
Alright.
Alright. Later.